this is going to go back to kind of cell biology, right? It's going to be a nucleus that holds DNA, right?
It's sometimes called the soma.
Correct. So the soma is what we call the body of the neurons. When we see them, am I cool to erase this real quick?
Yeah.
All right, cool. So the soma is going to be the body of the neuron, right? That's where we're going to hold most of the information and all of the organelles or the many pieces of the neuron. So when we look at the neuron, kind of shaped a little bit like this. We got this little long thing that sticks out, and then on the end it has some little fingers that kind of project. Right? Soma is going to be this bad boy here. The body.
That's the body, and that's the axon.
The axon.
And the dendrites are the smell.
Dendrites are the axon terminals down here. Cool.
Those are the ones that have that make the synaptic cleft.
Correct.
Good.
Yep. So, yeah. So you guys know more than you think you do. All right. So, we're gonna have a bunch of different organelles in here. We're gonna have things like mitochondria, right? Mitochondria is gonna produce ATP. I think we saw that a little bit later, so we won't get there yet. But the job of this neuron, or the purpose of the neuron, is to fire. That's its only job. So what we have to do is we always have to keep a neuron in a state where it's charged and ready to fire. All right? So typically how we do that is we keep the cell slightly negative on the inside. The resting membrane potential, or how negative we typically keep the inside of this cell, is negative 70 millivolts. That's how negative we like to keep the inside of this cell. And we do that by filling it with potassium. We shove it full of potassium, and we exclude or we keep sodium on the outside. And by separating these ions, we create a charge. When we separate them, they want to be together, and they want to be where the other one isn't. And so what we do is we end up creating a negative charge. So what happens is if we want to fire this neuron, we're going to open channels that let sodium in to make it more positive. As the cell becomes more positive, it's going to shoot up, and we're going to get what we call an action potential. That action potential is when the cell fires, or when the neuron fires. When the cell fires, we call that the action potential. Okay? And when that happens, we're going to get depolarization. So, meaning it's going to go from negative 70, where it's in a polarized state, and it's going to flip to a depolarized state, which is that positive 30 that you mentioned. When the cell flip-flops and it no longer stays polarized, we're going to shoot an electric current down the axon this way. And when you have a bunch of neurons kind of lined up in sequence, then we end up transmitting something down the course of a nerve. Any questions there?
Okay, the resting membrane is negative 70. When it fires, and is that what it's called, the depolarizing?
Yes. Yep.
So
So
I need to know, you need to know what hits then, and then when it, each is what it is then? The depolarization.
So typically what's going to happen is this cell is going to hover about right here, about negative 70. It's where it rests, right? Its baseline kind of idling sits at about negative 70. Not perfectly. It'll fluctuate a little bit, but it's going to sit about there. What's going to happen is as we start flooding this cell with sodium, it's slowly going to become more and more positive. As we flood this cell with sodium, we come closer and closer to something that we call our threshold potential, which is this negative 55. Threshold is that moment where we can't go. It's like the point of no return. If I become more and more positive, the second I hit negative 55 millivolts, all of the sodium channels open. Here I can open a few of them, maybe a few more. The second I hit negative 55 millivolts, everything opens. And I get a huge spike of sodium influx. When sodium comes in at that rate, it's going to depolarize. This is what we call a depolarization. Depolarize, a depolarization. So when it depolarizes, right, the idea of polarization is that it's opposites. I have two sides. So when I am polarized, I have potassium on one side, sodium on the other side, and they're split. They can't go to the other side. When I depolarize, now I've lost any control of sodium and potassium. So it's going to become polarized, or depolarized, excuse me. So it depolarizes. Now I have to repolarize it, right? We used the example last week of like those little cars that if you like pull them back, they'll like click, 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 and you can feel kind of that stored energy. And then when you let it go, it'll move. So it's kind of that same idea that right here where it's kind of in its locked position, it's ready to go. The second I hit negative 55, I let it go. It's going to run its course. Well, now the issue is it's moved down here. What do I have to do now? I have to reload it. I got to pull it back. So that's what I have to do here. That's what we call our repolarization phase. And it looks something like this. Repolarization is done by the sodium-potassium pump. The sodium-potassium pump is what restores the balance of the cell's membrane potential. Because the idea is that all of the sodium has flooded into the cell, a lot of the potassium has flown out of the cell. Now I have to put them back in their places. I have to tell all the sodium, hey, I want you out. Potassium, I want you in. And the job of the sodium-potassium pump, put it right here maybe. There's a bunch of them. They're all over. There's not just one. But the job of the sodium-potassium pump is to grab sodium, pull it out, grabs potassium, pull it back in. And it's going to do that. Boom, boom, boom. Right? And it's going to pull 3 sodium out for every 2 potassium it pulls in. I swear I can write this. So for every 3 potassium it pulls, 2 potassium it pulls in, it'll push 3 sodium out. So as it reestablishes its polarized state, that sodium-potassium pump usually is kind of it's an extra hard worker. And it's not going to bring it down to negative 70 where we usually sit. It actually sometimes goes a little too far and pulls it down to negative 90. That's what we call hyperpolarization. So, hyperpolarization only lasts a second. It's pretty short. And then slowly that cell is going to reestablish itself to negative 70. So this is what we call the action potential, right? So this is going to be super critical moving through the program and understanding how a lot of these work. Because this is how neurons fire, right? All of the neurons that we have in our muscles, in our joints, in our proprioceptors, this is how we get them to fire when we deliver something like adjustment. This has become super critical when we look at different ions or different supplements and how they affect the heart, for example, right? So this is super critical. Definitely know that. If you feel like maybe you're struggling with it, there's a lot of YouTube videos that are going to be really helpful. Just by briefly kind of going over that, it's super helpful. Okay, so on this study guide, there's a couple different things that it mentions. One is called the absolute refractory period. So an absolute refractory period is a time when the neuron cannot fire. Absolutely cannot fire again. And that is this phase right here. If you look at, let's use the example of that toy car. if I pull the toy car back, I let it go, and now it's down here, can this car move again right now? No, what does it have to do first? It has to be reloaded, right? Or if it's halfway through its journey, can I fire it again? No, it's already going. So an absolute refractory period happens in this time frame, where the cell has fired, and I can't get it to fire again. It won't happen. Because it's not loaded, it's not polarized, and it's not a state where I can actually fire again.
It's called the absolute refractory
The
period.
absolute refractory period. Cannot do it. Now, we do have another phase. If we have an absolute refractory period, we have a relative refractory period. So here's our absolute. Basically from here to here. I can't fire it again. It's already positive. The cell can't be fired. It can't be loaded again. But once it goes back below threshold, could I fire it again? Yeah. If it's below threshold, at that point, I can bring it back up to threshold and fire it again. Now, the issue is sometimes if it's in a hyperpolarized state, let's say down at -90, I have to bring it so much more positive compared to where I usually do. If it's normally sitting at -70, I only have to bring it up maybe 15 millivolts. But if I'm at -90, now I have to bring it up 35? I don't know math. Yeah, 35. So it can be harder, but it's still possible. This is what we call the relative refractory period. Okay. We should talk about refractory periods. Graded potentials. Graded potentials are these bad boys right here. These little dips, these little peaks and valleys, where they go up and down, but never pass -55. So I can have potentials that go up and down, but never cause the cell to fire. Those are what we call graded, because there's different grades. They can go up a little bit, they can go up a lot. As long as they don't hit -55, I'm good. Those are what we call graded potentials. Cool. How do we feel with that?
Fantastic.
Okay.
How do you feel?
Yeah? What questions do you have?
No,
I know you weren't here last week. This is a lot of what we went over last week. So, okay. So, moving through, I'm literally just going down the study guide. So, a couple things. So, membrane properties. So, with diffusion is going to be something where, let me write these out. So, we have a couple different ways that we can get ions across a cell membrane, right? If I want to get sodium into a cell or potassium out of a cell or vice versa, glucose into a cell or anything like that, in order to take them in, I have to get them across the cell membrane. There's a couple ways that I can do that. The first one is called diffusion. Diffusion is the easiest, most simple way to get things in and out of cells. Meaning it doesn't take any energy, it doesn't require any kind of special transporter. With diffusion, things just kind of flow into the cell or flow out of the cell. We don't have a lot of passive diffusion in the body. We don't like things to just kind of go wherever they want. We typically like to control how things get in and out of cells. So that's where we create something called facilitated transport, which is where we have maybe a specific receptor or a specific channel that lets things in and out of cells, just so we can control them. So let's say I have a sodium channel that's specific for sodium, so I can let sodium in and out of the cell. Perfect. Or a potassium channel, or a glucose transporter. All of those things are going to facilitate the transport of something across the cell membrane. Typically with facilitated transport, it does not require ATP. So same as diffusion, it doesn't require ATP, it doesn't require any energy. But it only happens at specific gates or specific channels, versus diffusion can kind of just happen anywhere. Things just flow in and out where they want to go. The big one, the really important one, is something that's called active transport. One thing with these is that diffusion and facilitated transport only go down the concentration gradient. That means that something will only move from an area of high concentration to low concentration. If I have a hill, can I roll a ball up a hill? No, it won't roll up. I have to push it up, of course, but it's not going to passively just roll up a hill. It will only roll down. It's the same thing with diffusion. It's only going to go from high concentration down to a low concentration. Active transport is the opposite. With active transport, I can push the ball up the hill to where I want it to go. But that doesn't happen passively. Active transport requires ATP. That's what you need. So active transport requires ATP, requires energy. The big active transport that we've talked about right now is this guy, the sodium-potassium pump. The sodium-potassium pump puts things where they don't want to go. It's going to move them against their concentration gradients. So that's going to be a big thing. Why neurons need so much ATP? Why neurons need glucose? That's because they constantly have to fire. They have these little engines running all the time, trying to keep things in their place. Right? So active transport, it's sometimes known as also primary active transport. Is a word you might hear. That's the big boy here. That's the big one to know. Primary active transport requires ATP. Cool. There's other things too. There's like secondary active transport, which we're not going to really. Has he talked about that at all?
No.
No. Okay. It's maybe mentioned, or it used to be mentioned in the third quarter class, visceral physiology, but they'll talk about it next quarter. But at least you know active transport requires ATP. Everything else does not. All right. Okay. Summation types. So there's a couple ways that we can get this neuron to fire. This is what we talked about last week. So let's say, in order to get this neuron, in order to get this neuron to fire, I have to get it to what percent, or what electrical charge? Where's our threshold? Negative 55, right? So if I'm sitting at negative 70, what I have to do is I have to get enough sodium into this cell. I have to send enough of a spark in order to jump it to negative 55. Sometimes, let's imagine there's another neuron here. Is that a hand?
No.
Oh, okay, sorry. Let's imagine there's another neuron here. And it has a couple connections leading to this guy right here. Okay? If I were to activate just this one right here. This is dendrite 1, this is dendrite 2. If I just activate dendrite 1, I'm going to let some sodium into the cell. And as sodium comes into the cell, what happens to the charge of this neuron here? If I were to measure it, yeah, it's going to go up. It's going to become more positive. But let's say only activating dendrite 1 isn't enough stimulus to get it to negative 55. Just with one dendrite, it's not going to get it there. Well, what if I fire 2 of them? What if both of these guys start flooding the cell with sodium? Well, now, instead of it just going up here and kind of pooping back out, it gets me to negative 55. This is what we call summation. So I now have 2 stimuli summating together, or adding together, in order to get me to negative 55. So there's a couple ways that I can do that. The way I explained this last week is, let's say, this is my neuron right here. I'm a neuron, and I'm trying to get this guy to fire. If I stimulate it I give it a shot. Boom. That's not enough to fire it. It needs actually 2. Okay? There's a couple ways that I can do that. Either I can shock it here and I can shock it here. Boom, boom. Now it's going to fire, right? It has 2 stimuli happening in space. 2 separate stimuli. Or what I can do is I'll take this hand, I have just 1, and I do it twice. Boom, boom. Now I've hit 2. So that was kind of the example that we gave last week, right? I can have 2 stimuli over time. Maybe number 1 sends it twice, and so we get that activation when we get here. Or I do it spatially, where I have 2 different stimuli happening at the same time. Okay? So we call temporal summation. Those are words that you might use on the test. Temporal summation over time, and spatial summation in space. So that's how we grade enough potential to get to -55. Either temporal summation in time, I hit kind of like that double jump in Mario. 1 jump isn't enough, I might need 2, boing boing. So I hit it boom boom 2 times over time. Or if I needed 2 stimuli, maybe I can have 2 different stimuli at the same time in space. Spatial summation. Okay. How do we feel with summation? Awesome. I'm gonna see real quick. Alright, sweet. So then if we go through the neurotransmitters, a lot of them are related to their name. So you have here things like cholinergic neurons. Cholinergic relates to acetylcholine. We're going to talk a little bit about those effects. Things like GABA or glutamate. Those are all different neurotransmitters that have different effects, and we'll get into some of those. But, okay. Lastly, motor neurons. We have alpha motor neurons and gamma motor neurons. Okay.
It's almost like he's
So, yes. And since we're just going through it all super fast, it doesn't help either. All right.
It does help. This is fantastic.
Good, good, good. So, if I want to contract, let's say my quad, I'm going to contract my quad. I have to move it, right? So what we have is we have an alpha motor neuron. The alpha motor neuron is what tells my quad, the beefy part of my muscle, to contract. The alpha motor neuron is what gets the muscle to contract. So when we talk about like, oh, your femoral nerve, right? You guys go through like gross anatomy right now?
Yes.
Let's go gross anatomy. So femoral nerve innervates my quads. It's going to tell my quad to contract. That's going to be an alpha motor neuron. At the same time,
So the alpha motor are for muscles.
correct. The alpha motor neuron,
And gamma is for smooth muscle, isn't it? Or am I?
not quite. So, I'm trying to get a way to show it. Okay. Let's imagine that this is my muscle here. This will be way better than me actually drawing it out. If we take a look at this muscle right here, if I were to, this is the big red part, right? That's going to be the bulk of the muscle. That's what the alpha motor neuron is innervating, the big bulky part of the muscle that's going to get to contract. Now, deep inside of the muscle, you're going to see these 2 little structures here. These are what we call muscle spindles. And their whole job is they're like little undercover muscle fibers. And their job is to report back to the brain about how lengthened the muscle is.
You're referring to the red spiral?
I'm talking to the little red spiral. Yep. So, here's an example. If I were to close my eyes and contract my arm, I can't see my arm, but I can tell that my arm is sitting at about maybe 90 degrees contracted, right? You can do that with the rest of your body. That's the reason you can walk with your eyes closed. You know where your body is in space, and you know how lengthened your muscles are, even though you can't see them. Those are your muscle spindles telling your brain, hey, I'm lengthened, hey, I'm shortened. And their whole job is to just tell the brain how lengthened or shortened the muscle is. Okay? They're like little undercover muscle fibers. The problem is they are not muscle tissue. They're neural tissue. They're sensors. They're not muscle. So what we have to do, now I'm going to draw this out. Am I cool to erase this big mess here? This is the NeuroFits crash course. So, let's imagine this is my muscle. On the inside of that, I have these muscle spindles, right? But if you imagine that, if the muscle contracts or shortens, this isn't connected to anything. It's just kind of in the middle of the muscle. How do I get this? Imagine it as like a little spring, and the spring is what tells the brain how stretched or how shortened it is. That's a good way of putting it. Well, the problem is if I just stick a spring on the inside of my muscle, I have to connect it on both sides in order to stretch it or contract it. Okay? So what I have is I actually have little muscles attached to the end of the muscle spindle. And these little muscles on the inside are what we call intrafusal fibers. Versus these big ones, we talk about the big part of the muscle, the big meaty part. These are what we call extrafusal fibers. What type of motor neurons innervate the extrafusal fibers? Those are the ones we've talked about so far. Yeah, these are the alpha motor neurons. Innervate the extrafusal fibers. These are the ones moving the muscle. But the intrafusal fibers are connected to the muscle spindles. Those little neurons, the little springs on the inside that tell it if it's lengthened or shortened. And the thing is I have to contract or stretch these at the same time as I shorten and lengthen the rest of my muscle. This seems super counterintuitive, but they have to contract at the same time in order for me to pull and stretch the spring as the muscle lengthens and shortens. So at the same time as I have my alpha motor neurons innervating the big extrafusal fibers, I also have motor neurons contracting these little intrafusal fibers. These are what we call a gamma motor neuron. So, gamma motor neurons and alpha motor neurons contract at the same time. Alpha motor neurons move it. Gamma motor neurons send information up to the brain. Now, this can be a problem. Let's say for some reason your gamma motor neurons start firing when your alpha motor neurons are not. What is your brain going to think is happening with this muscle? It's going to think it's contracted. If I fire my gamma motor neurons but not my alpha motor neurons, they're just chilling here, but my brain is saying, oh, it feels tight in here. How is that going to feel for the person? Bad. It's going to feel tight. So this is a mechanism by which we believe, it's still a theory, by which things like low back pain or muscle tightness or muscle spasms occur, is that we have discoordination of alpha motor neurons and gamma motor neurons at the same time. And so what we can do is we can stretch these muscles, maybe using something like an adjustment, and we can actually reset the alpha-gamma contraction. That's irrelevant to the class, that's just kind of my own thing, but the alpha-gamma co-contraction theory is one of the things that says muscles might just be tight because they're tight for no reason. And so a lot of times we can stretch a muscle using an adjustment, and a lot of times somebody's like, wow, my muscle's not tight anymore. My low back feels so much better. How'd you do that? You didn't do anything besides just stretching the muscle with an adjustment. This is how physiologically it makes sense. Cool stuff. Chiropractic works. Okay, so those are what we call our alpha and our gamma motor neurons. What do the muscle spindles tell the brain? These little muscle spindles here tell the brain about the length of muscle. They don't tell it how hard it's contracting or if there's any stretch, but it just says, hey, lengthen, hey, I'm short. Okay. Moving on to week 2, skeletal muscle. How do you want to start this? So, muscles, let me pull up the PowerPoints before I start going off the rails with the exercise physiology type stuff. Okay, so when our neurons talk to each other, they talk to each other across something called a synapse, right? The synapse is the space between two neurons. If neuron 1 wants to talk to neuron 2, they don't actually touch each other and communicate. They basically will almost touch, and there's a little space between the two of them. That's what we call the synapse. And across those, we send little chemical connections, right? This one's going to be electrical. The synapse is going to be chemical. We're going to send some kind of neurotransmitter between the two of them. And then this is going to start another electrical signal. Okay, these are all just muscle contraction. Cool. Let's go through this real fast. Am I cool to erase this?
Yes.
Awesome. It's engineers are kind of warm in here.
This place is a little warm.
I'm a little chilly.
Really?
I
Okay.
have an ice drink,
I was like,
maybe that's why.
sweating.
Oh no. The pressure's maybe on you.
Yeah. All right. So when we look at muscle contraction,
I opened the door for a little bit of cold air. I can close that. I didn't realize they had
I don't want you to sweat.
no, it's all good. It's all good. Okay, here we have our muscle. Here we have our neuron. We're going to call this the motor neuron. So we have our motor neuron and our muscle. This is something that we call the motor end plate, or the connection where the motor neuron meets the muscle. So, motor neuron is going to send a signal across this synapse, right? Is that space between two neurons. In this case, it's a muscle, but we're still going to call it a synapse. We actually call this the synaptic cleft, just a more specific name. When a neuron meets a muscle, we call it the synaptic cleft. I'm going to go through this kind of fast. Stop me at any point if it's too much. So, what is the ion? Well, let's backtrack just a little bit. We talked about action potentials. If I'm getting a motor neuron to fire, what is the ion responsible for triggering the depolarization? Or as I send an action potential, which ion is depolarizing the nerve? The motor nerve. What ion is it? We've got sodium, potassium, we've got chloride, we've got glucose, we've got all kinds of different things. What's triggering the depolarization of this nerve?
Sodium.
Yeah, it's sodium. Okay. As sodium from the outside is coming into the cell, it's becoming more and more and more positive. So sodium is what's causing the depolarization of the nerve. Now eventually it's going to reach the end of the nerve. Here's the end of the road. This is as far as that depolarization is going to go. The question is, how are we now going to send a signal across this space? Well, that's what we're going to go over right now. So as sodium makes it to the end here, step 1, sodium. Sodium's coming in. We already know that. It started at the cell body way up there. It's going all the way down to this point. Step 2. As it becomes more positive, calcium is now going to flow into the cell. The influx of sodium will trigger the influx of calcium. Right? And these, they don't just flow in. Calcium doesn't just like walk into the cell. We have calcium channels, right? We talked about facilitated diffusion, how there are specific channels where things have to come in and out. They don't just diffuse into the cell. So here we have little calcium channels that are going to open. These are sodium channels up here. So as the cell becomes more and more positive, these calcium channels open, letting calcium into the cell. Okay. How do we feel with that? Sodium first, calcium next, at the very end. Calcium's like almost the second to last step. Awesome. Well, what do we need to cross this, right? This is all electrical. What kind of transmission has to occur across the synapse? I said that earlier. It has to be a chemical. So what we're going to do is we're going to release a neurotransmitter across this. A neurotransmitter is what tells a nerve to fire, to be quiet, to activate, right? It kind of is how it gives its instructions to the other nerves around it. So in order to activate the motor nerve, in order to send a signal to activate the muscle, to tell the, let's say this is the bicep, to tell the bicep to contract, this motor nerve has to send some kind of neurotransmitter to activate it. In muscle contraction, in any motor movement, the neurotransmitter is acetylcholine. That is the neurotransmitter associated with muscle contraction. That's going to be huge to know. Any muscle movement is controlled by acetylcholine, or is activated by acetylcholine. So if a nerve connected to a muscle releases acetylcholine, assuming everything is working properly, that's going to trigger a muscle to fire. So, where does this acetylcholine come from? If we're going to send acetylcholine to the muscle, what we have these little bubbles. They're filled with acetylcholine molecules. And they're just chilling here, waiting to send their acetylcholine to the muscle. And the way that these guys will release acetylcholine, because they're just like sitting in there waiting, they're just little bubbles filled with acetylcholine. In order to get this nerve to spit out that acetylcholine, I need calcium. Calcium is what triggers, and this is an old test question from the previous version of the course. I don't know if you'll use it, but write it down just in case. They might ask something. I don't know if he's changed the questions, but I'm going to give it to you guys just in case anyways. The question went something like this. Which ion will trigger the release of a neurotransmitter? Something along those lines. Or a neurotransmitter release is modulated by which ion? However they want to ask it. But they're going to be relating the release of a neurotransmitter to some ion activation. And a lot of times people will say, their reasoning will be, well, sodium is what depolarizes the nerve, so sodium must be the one in charge, must be the one responsible. That's not bad thinking, because sodium was step 1. Sodium did do a lot of that. But the release of the neurotransmitter is directly controlled by calcium coming into the cell. So the correct answer in that case would be calcium. Calcium is what causes the release of a neurotransmitter, not directly sodium. Does that make sense? So it's like A equals B equals C, right? But he's not asking if does A equal C, it's B equals C. Stupid. Okay. I'm going off the rails here. But if he uses any one of those questions, it's calcium.
Given the context, though, it made sense.
Okay, cool, awesome. So what's going to happen is as soon as calcium comes in, Calcium tells these bubbles, hey, you're good. Go release them. If anything, we call these synaptic vesicles. If you hear those terms. I'm going to use as many terms as possible just so that he's not going to throw stuff out there and confuse you and you're like, my gosh, Dr. Mann, what the heck is a synaptic vesicle? And it's like, oh, it's these little bubbles. You're like, can't you just say that? So, synaptic vesicles, they release the neurotransmitter, and the way that they're going to do that is they're going to kind of migrate. They kind of hang out in this general area. As the calcium comes in, they slowly start migrating down to the edge of the cell. But what they're going to do is they're actually going to fuse with the cell membrane. You kind of see how that is. They're going to like come all the way down until they fuse with the cell membrane. And then that neurotransmitter gets spilled out into the synaptic space, the synaptic cleft. Does that visual kind of make sense there? Mm-hmm. Cool. So this is what neurotransmitter is this right here in the synaptic cleft?
Acetylcholine.
Acetylcholine, yeah. So acetylcholine is then going to bind to an acetylcholine receptor on the other side. That is what we call a cholinergic receptor. Cholinergic because it receives what neurotransmitter? Acetylcholine. That can be tricky. A lot of times people will hear those words and they're like, cholinergic, is it epinephrine? It's like, oh, it's the one in the name, acetylcholine. Okay? We have these little receptors on the motor end plate, and they receive that acetylcholine. An acetylcholine binding to the muscle is what starts the whole process of muscle contraction. Now we have activation of the muscle. Did he go all the way into depth of things like actin and myosin and tropomyosin and all of that muscle stuff?
Briefly.
Okay, cool. We'll fly through that. Do you guys want me to go through that, or would you rather just like watch a video on it?
Watch a video on it.
To go over it? Okay, awesome. We can go over it. I just don't want to be like explaining stuff if you guys are like, this isn't like easy to get at things. Cool. So the same way, we're going to go through a couple steps pretty fast. The same way the nerve, when it receives a neurotransmitter, lets in sodium, kind of starts that process. When acetylcholine binds to the motor end plate, we also let sodium into the muscle now.
So would that be like step 4?
Sure. Yeah. So we'll number these. So this was step 3. We get released the synaptic vesicles. Now sodium comes in. Yep. Okay. How do we feel with that process? Sodium comes in, triggers calcium to come in. Calcium triggers the release of the neurotransmitter. The neurotransmitter then lets sodium into the muscle. Boom, boom, boom. Okay. Now, let's move on. Zoom in. We're no longer going to focus on the motor neuron. The motor neuron did its job. At that point, the motor neuron's like, here's your acetylcholine fire muscle. And then it just kind of like relaxes, because now the muscle's in charge. The muscle's now going to contract. So, all right, we'll do a thing real quick. Okay, quick thing as well. As long as this acetylcholine is stuck to one of these receptors, it's kind of like pressing the on button, right? If I press the on button, sodium starts flooding in. The second I let go of the on button, the sodium stops. Okay? It's kind of the same way. It's like if I touch them together, the acetylcholine and the receptor, like it starts going. The problem is if I never start, or if I contract a muscle and I never stop activating the muscle, let's say the muscle fires, what's going to happen if I never stop stimulating the muscle? It's never going to relax. Maybe a tricky question. If I'm constantly sending acetylcholine all the time to a muscle, and I don't ever stop sending acetylcholine, that muscle is never going to stop. It's like, okay, cool, when are you going to stop sending me acetylcholine? I'm still contracted. That's a problem, right? So, this can be an issue. There's a disease. Have you guys ever heard of tetanus? Yeah? So, tetanus, what does it do to a person? Do you guys know?
It locks up the muscles.
Yeah, it locks up the muscles. So that's why it's sometimes called like lockjaw, right? People that their muscles stay contracted and they can't stop. Or there's like pretty awful paintings from like back in the day where their whole bodies will contract and they'll get stiff, right? Everything's contracted and they can't move. The reason that happens is because we have constant activation of the motor nerve, because we're missing the enzyme that breaks this up and says to the muscle, okay, you can relax now. You did your job. Thank you. Let's reset. If they don't have that enzyme that breaks it down, the muscle never relaxes. That enzyme is called acetylcholine esterase. Esterase. Acetylcholine esterase. ACE is the name of a cleaving enzyme, right? It breaks things down. So acetylcholine esterase is going to break down acetylcholine after it activates the muscle. And that happens normally. So it's kind of like acetylcholine is going to say, hey, muscle, fire, 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 fire. And then acetylcholine esterase is going to say, okay, that's enough. That's enough. Okay, go back home. Leave the muscle alone. Let it relax again. And that's a normal process. That's how I can contract my bicep and then relax it right after. Think about how often that has to happen in something like walking or running, right? A lot of muscles have to contract and then relax, and contract and relax, and it's like a really complicated game. So this is super critical and super important for acetylcholine esterase to also be able to break it up super fast and then reset the muscle for contraction again. So, acetylcholine is going to get broken down into two different molecules called acetate and choline, and then it gets recycled. And then we just reform acetylcholine over and over again as needed.
This is go till 1? Oh, wow.
Yeah. So I'm here as long as you guys want to be here. You guys don't have to stay the full time. You guys can always come late. I'm here from 11 to 1. So every week. Okay. So let's call that good. This is also, this whole diagram that we called here is something called the neuromuscular junction, or the connection between the neuron and the muscle. Sometimes referred to as the neuromuscular junction, if you see that. Any questions with that? We feel pretty good with that? Pretty solid?
So far, so good, yes.
Okay. So we have a couple of different types of muscles in our body. He has here muscle fiber types. So our type 1 muscle fibers are going to be what we call our slow twitch fibers. So type 1 fibers are going to be the ones that fatigue very slowly. These are the ones that tend to, like our postural muscles, for example. They will constantly stay contracted and hold for a long time. They don't get tired easily. So these are also ones that like help us with, like if I were to run or jog at a very easy pace, those muscles don't get tired fast. You don't get sore from a really slow jog. Whereas my type 2 muscle fibers tend to fatigue very quickly. That's going to be like if I were to run a sprint, they're going to get sore. Or if I was to do like a powerlifting move, they contract very quickly and then they fatigue very quickly. So the primary fuel source for our type 2 fibers is going to be glycogen and glucose, which is what he has here. So I'll just go through those. So type 1 slow twitch fibers are slow to fatigue. Our type 2 muscle fibers, or our fast twitch fibers is sometimes what we call them, fatigue quickly. They burn up glucose really fast and then they get tired. And then our type 2B fibers are going to be kind of more of a mixed fiber. It has a little combination of the two. Okay. let's get into the actual muscle contraction. This is like so all over the place. What is the job of acetylcholinesterase?
To relax the muscle.
Yes.
And then get it repaired again.
Perfect.
Right.
Yeah. And what does it do exactly? It destroys acetylcholine. It cleaves acetylcholine. Sweet. Okay. Let me pull up a picture of. Way better than me trying to draw this out. I did not go to art school. Hey, hey.
It's muscle cooling inside. Right.
Alright, so now we're going to be going over something. So we're going over the midterm study guide that he put on Blackboard.
Sweet.
We're just going through that, making sure that everyone kind of caught up to speed with everything that could potentially be on the test next week.
Okay.
Right now we're in week 2. We're going over sliding filament theory. So, the sliding filament theory. So we've talked about how we're going to get this muscle to activate, right? Calcium triggers the release of a neurotransmitter. The neurotransmitter binding causes sodium to flood into the cell and start the process for contraction. Okay. Now, let's go step 5. Step 5. Step 4 was what? All this sodium coming into the cell. Let's imagine deep inside of this cell, I have these big tanks. These big tanks are filled with calcium. These are like big gas tanks. This is something that we call the sarcoplasmic reticulum, if you heard that before. Sarcoplasmic reticulum is a big tank filled with calcium. So after we start flooding as we start flooding the muscle with sodium, sodium is going to cause these little tanks, they're not little, they're big, these big tanks of calcium to release the calcium into the muscle. Because it's kind of stored. The calcium is stored inside the muscle, inside of the sarcoplasmic reticulum. Sodium, through a long chain of events we're not going to go into, is going to release calcium. It's going to flood the cell, the muscle fiber, with calcium. That's step 5. We'll draw it over here. Calcium flooding the inside of the cell. I know this looks like a total disaster. There's like arrows pointing in every which way. So we'll go through those steps one more time. Sodium triggered the depolarization of the motor nerve, way up at the cell body. It's going to set an action potential all the way down till we get here. After the cell is slowly depolarizing, that triggers the influx of calcium. Calcium is what actually ends up causing the release of the neurotransmitter. When the neurotransmitter, which in this case is acetylcholine, the one related to muscle contraction, acetylcholine binds to the motor end plate, causing sodium to come into the cell, which lastly triggers the sarcoplasmic reticulum, to release, flooding the muscle cell with calcium. That's where we're at.
So 3 is the release of, you said acetylcholine. What's the ACH esterase?
So acetylcholinesterase is the enzyme that breaks down acetylcholine after it's activated the muscle.
Gotcha.
We give the example of tetanus or tetany, like the disease. Tetany gets rid of acetylcholinesterase. And so the muscle never relaxes. It no longer can stop firing. It is constantly activated by acetylcholine because it's never broken down. So the person stays contracted, their jaws locked, their muscles stay contracted, and they can't relax because they don't have acetylcholinesterase.
Yeah, you can't make it anymore.
Yep. Correct.
4 is not the is it the repolarization?
Which one?
4.
4 is the depolarization of the muscle. We have sodium flooding into the muscle. Yeah, absolutely. Before you guys go, I don't have any paper. No,
I have paper.
I have paper, right? It's fine. Just so I can get everybody's name on here.
And then you said 5 is when calcium is leaving the sarcoplasm.
This is neuro. Yeah, absolutely. Best of luck next week. Cool. So, calcium is now flooding into the cell. That's going to start the process of muscle contraction. I'm not going to draw this out. I'm going to pull it up on my iPad because I am not a good artist. So, I'll walk around. I'll kind of make this clear. So, we have a couple little strings, like little chains wrapped around each other, right? We have this green chain, and it has this little blue chain that covers it. You can see on these like little green beads that they have like a little tip on them that's like a dark green. You guys see that? The way I like to think of them is kind of the way like if you look at an olive, right? It's got like a little hole that's been punched out at the top. It's kind of the same way. It's like, I don't know if you guys were like this as a kid, but what do you want to do with the olive that has a hole in it as a kid?
Okay.
You're going to put it on your fingers, right? And it's kind of the same way. So, these are like little olives that have a little binding site at the tip. And we have this little light blue chain that actually sits right across all of those binding sites and covers it up. Nothing can bind to it, nothing can stick to it, because that little chain is covering it. Okay?
Yeah.
This green thing is what we call actin. Actin is the green one. And actin has a long time complicated relationship with this purple thing down here called myosin. And actin and myosin want to be together. And if you see here, the myosin head kind of looks like a little finger, and the little binding site on actin kind of looks like the whole inside of a olive. And that myosin head wants to stick to the little binding site on actin. Okay? But we have this little guy that's always in the way. He's always getting in the way of actin and myosin being together. This is a guy that we call tropomyosin. Tropomyosin is this blue chain. So tropomyosin is constantly blocking actin and myosin being together. One more thing before we go there. I know there's like 8,000 words, but I'm just like naming off. In order to get tropomyosin to move out of the way, he's like that little kid. Yeah, I don't know. I'm not gonna go there.
Like a little brother in the way when you come
Yeah, okay, that's a better example. Yeah, so, you know, older sister and her boyfriend just want to be together, and you got that snot-nosed little brother that's in the way. So the only way that you can get tropomyosin out of the way so that actin and myosin can be together is to grab it by its little troponin. Troponin is a little protein on it. And what you have to do is you have to yank troponin in order to move tropomyosin. Okay? You can see it's like these little blue pieces, these little blue bulbs. That is troponin.
So you said troponin is like the little brother?
Yeah. So I'll go through everything one more time, just so we're kind of aware. Actin is the green, myosin is the purple. Green is the sister.
Yep.
Myosin is the boyfriend.
Yep.
Okay. The snot-nosed little brother that's for lack of a better word, kind of cock blocking, right? I didn't want to use the word, but we'll use it. Right? The little brother is this blue chain. That is tropomyosin. Tropomyosin is the snot-nosed little brother that won't let actin and myosin get together. The only way that tropomyosin will move out of the way is if you put his troponin and calcium together.
Gotta give him a little treat to go away.
Sure. You give him a little treat and he goes into the other room and now his sister and her boyfriend can be together. Yeah. So, the way that we're going to do that is we're going to flood the cell full of calcium. Calcium and troponin have a long-time relationship. This is just a complicated story. Troponin and calcium want to be together. When calcium floods this whole cell, it's going to take troponin and it's going to move it out of the way. So when troponin moves out of the way with calcium, it's going to drag tropomyosin with it. And so this little blue chain now pulls and exposes all of those binding sites on actin. Great. Well, now actin and myosin now have all of their binding sites open to be with each other. And so what's going to happen is you get, and that's why we have like that's step 1. We have 2, 3, and 4. What's going to happen is that now you can see troponin has moved out of the way, and all these binding sites are now available.
Mm-hmm.
Myosin is going to stick itself. It's going to go and stick itself to actin. And then what it's going to do is it's going to flex. So you can see that it's gone from this position to this position. Okay? And what happens is if I imagine crawling, the way crawling kind of looks. What's going to happen is we don't just have like one actin or one myosin, we have like millions of them. And what they're going to do is if we expose the binding sites on actin, myosin is going to stick itself, it's going to contract, it's going to release, it's going to stick itself, and contract. What you're going to get is this kind of mechanism that looks like this. If it happens at a really fast pace.
Almost like monkey bars.
Kind of, yeah. It's going to go. And what you're going to get is you're going to get these two filaments, actin and myosin, that are going to slide past each other like this. They're going to go. And what happens is if we stick a bunch of those in sequence, and if you imagine that in a very, very molecular, if you look down deep into the muscle, what those are going to do is they're going to contract and they're going to relax. And that's the way that a muscle will contract and it'll relax. Okay. That's the very quick condensed version of it. I mean, in undergrad, I had a whole class that just went over this for a whole quarter, and you guys got it in 20 minutes.
So,
It's like a wrench, right, where it goes back and forth.
Yes, correct.
So
Mm-hmm.
so for the relaxation, that's passive. It doesn't move the actin and myosin, correct?
Correct.
It's just this, like,
Correct.
slap.
It's the actual contraction that requires
That's
ATP.
what I thought.
Mm-hmm. Okay, next step. So now that we've got kind of the mechanism that they're going to... That requires ATP. That myosin head is going to stick itself, but it's not going to just contract by itself. It requires energy in order to do that. So what happens is we have this ATP. I think we're going to go here. Okay. Let's say it's chilling here. In order to reload it, like let's say you're gonna like reload a pistol, right? A pistol's not just gonna like fire over and over again. You have to like pull back that little piece, right? To like get it to go back, right? So it's kind of the same way. ATP has like fired its shot, like it's done. So what do I have to do in order to get it back? Is I have to break it down in order to get. I have to phosphorylate ATP in order to get it ready to go. So in order to get it from this position to this position where it's ready to go, I have to turn ATP into ADP. Alright? How comfortable do we feel with that? Phosphorylation?
bringing it back.
Bringing it back. We're reloading the myosin head. So we phosphorylate it, we turn adenosine triphosphate into adenosine diphosphate and an extra inorganic phosphate. That allows the myosin head to restick itself. It allows it to contract, But in order to release it, I have to stick on another ATP. It requires energy. That's why exercise requires so much energy in order to get your muscles to contract, because we start using ATP. This is where muscles get their glucose from. This is why.
We said it's a dephosphorylation, it's losing phosphorus.
Correct, yes, sorry.
No, it's okay, I struggle with that.
So what about the for the dephosphorylated?
The contraction, or the, better yet, the reloading of the myosin head. So that is what we call the sliding filament theory. It's still a theory because we technically haven't proven it. We can't see these guys. They're too small for us to really see, but we have evidence to show that this is probably how this works. Kind of crazy. And we teach it as if it were not a theory, but it technically still is.
So we need to move the troponin to expose the tropomyosin to actin. No, tropomyosin is the blocker.
Yep.
So we want myosin and, what was it, actin?
Yep. Actin is the sister, myosin is the boyfriend.
Okay.
So the way that I like to remember it, and it actually doesn't work this way with the way it's spelled at all, but calcium turns troponin on. It's spelled troponin in.
Okay.
But the way I just memorized it in my head is like troponin on. Troponin on. Versus tropomyosin. Since they both start with tropo, it's kind of like, which one's which? Troponin on. And that's in. I have a lot of weird things like that that kind of bounce around in my head, but works for me. Hopefully it works for you. So that is what we call the power stroke. The power stroke is the myosin head is contracting. That power stroke. Just in case you hear that. So we have a couple types of muscle contraction. One of them is called concentric. Concentric is what we think of as the contracting phase. So if you think about me holding a weight, and if I were to do a bicep curl against gravity, my muscle is shortening, it's contracting, so we call concentric muscle contraction. Now, if I were to completely release my muscle, it would just fall back down. But ideally, I don't want to have a 40-pound weight in my hand and just like let it fall. That would be bad. So what do I do on the way down? I have to control it on the way down. It's still contracting, because if I were to completely relax it, it would fall. This is what we call an eccentric contraction. Concentric is a shortening contraction. Eccentric is a lengthening contraction. Okay?
Say concentric, shortening.
Shortening.
Eccentric.
Eccentric is a lengthening contraction. Sometimes, like in exercise, they might say like control the negative. Like if you're like, releasing a weight, that's the negative part of the lift. A couple other types of contractions. We have what we call isometric contractions. Iso meaning same, metric meaning length or distance. So what does an isometric contraction look like?
It's contracting, but it's not moving.
Correct. It's like a bodybuilding pose, right? I just stay here and I contract. It's not moving. That is a isometric contraction. A contraction with no movement. Versus an isotonic contraction. We don't really use a whole lot because you have to have a very specialized machine to do isotonic contractions. Those are ones that move at the same
It's like the same pressure.
it's the same muscle strength is required for the whole motion. So a lot of times we'll use these in like rehab settings, or you use it to test muscle strength. So a big one, like the most common one, is going to be, it's a leg extension. And so it looks like a common leg extension machine. You guys know, right? Like it'll go on your shin, right? You kind of lift it up. But usually with like a standard one, you can just kind of like lift it and let it go, and you can change how fast you push it up or how slow. With an isotonic contraction, no matter how hard you push, it's going to move at the same rate. It's going to move at the same rate the whole time. No matter how hard or how soft you push, it's always going to move at the same rate. Kind of tricky, but we don't use those a whole lot.
Would you say like rate or like resistance?
That's the word.
Okay.
Thank you. I was like, I know there's like a more official term for it. Cool. Alright, let's keep going. So we have a couple different energy systems, right? We get energy, this is just a flashback to freaking biochemistry, right? Glycolysis is gonna be our main source of energy. With glycolysis, what's our main substrate for glycolysis?
Okay.
Glucose. Glucose is gonna be our primary source of energy. And in the end, with glycolysis, how many ATP do we form?
2.
Yeah. I think we technically form 4, but then we use 2, right?
Oh
Is that
yeah,
how that
because
works?
of the,
Yeah.
I don't know, I'm trying to remember. I'm like, I don't know, it's been a minute.
it's like per glucose, yes.
Cool. So that's obviously going to feed into like then the citric acid cycle, and then we get into the other one, electron transport train, and then we get like a crap ton more. That's not important. So the other one is going to be our lactic acid system, right? Which is going to be our anaerobic pathway, where we end up producing a bunch of extra acids like lactic acid. This is what happens when our muscles don't have enough, speaking in terms of muscle, when our muscles don't have enough oxygen.
Mhm.
So this is going to be your anaerobic metabolism versus aerobic. It's going to be kind of like slow cardio is your aerobic, as long as you can breathe heavily. You shouldn't get sore with aerobics, but with anaerobic exercise, you should get sore.
This neuro.
Yes, ma'am. Cool.
Okay.
There you go. Is it laundry or laundry?
Somebody's sitting.
Yeah. I don't think so.
Alright, now let's talk about a couple pathologies. These are like all over the place.
Yeah. Let me make sure it's correct with him. I think it's right.
Yeah.
What's up? What kind
Just
of?
wanna make sure. Just like the steps of the sliding filament theory. So I have 1, move troponin needs CA2.
Mm-hmm.
2, troponin moves with the tropomyosin, which opens the binding sites.
Yep.
3, myosin binds to actin and moves down the sites.
Mm-hmm.
4, myosin reloads and relaxes through dephosphorylation.
Correct.
Yeah.
Cool.
I forgot my finger. Let's see this.
I'll let you know.
That was a good way. Put that down.
Alright, now let's go through a couple.
I can explain that to you too.
Thank you. I was gonna write it down, but I'm like, I need a minute to process that, so.
Alright, so let's talk about a couple diseases related to muscle contraction. So the first one is going to be something called myasthenia gravis. So myasthenia gravis is going to be a disorder of muscle contraction.
Can you spell it?
Um,
Can you write it?
so first disorder is going to be myasthenia gravis.
What's your name again?
My name's Chandler. Sorry, I'm gonna pull something up because there's two diseases here and I'm gonna be able to compare the two. 1 second.
I thought there was a third one in that lecture too, something about botulism.
Yeah, so there'll be a couple actually that we'll probably talk about. Botulism, tetanus, myasthenia gravis, and then Lambert-Eaton, which I haven't talked about Lambert-Eaton, so it's definitely a new one for this. But okay, so let's start with myasthenia gravis. So let's imagine that you have an autoimmune condition. And that autoimmune condition, what it's going to do is it is going to destroy, this is such a mess here. Let me just erase a couple things. I'm gonna drive myself crazy looking at this. Okay, I have these receptors on the muscle side. These receptors, what they're going to do is they're going to receive acetylcholine that's released by the nerve. In a normal person, these receptors are all nice and chill, ready to receive acetylcholine. And that's going to start the whole muscle contraction process. In myasthenia gravis, what happens is we get autoimmune destruction of these receptors. So if I don't have receptors, what happens to the muscle? It doesn't get any stimulation. It doesn't fire. So where we typically see myasthenia gravis more commonly is in the face. So what is it going to look like in the face of somebody who has myasthenia gravis?
Almost like a paralysis.
It's going to look like paralysis, or they're going to have really weak movement of their muscles. So they're going to get facial drooping. It's going to look kind of like a patient with something like a stroke.
So you said it destroys those little
It
receptors.
destroys the receptors. So you can see here, we're going to get autoimmune. Oh my gosh, I hate iPads sometimes. 1 sec.
That was a good picture.
Yeah, this is perfect. Sometimes these are going to explain it a lot better than I can.
Steve, and the pastor of the Baptist Church as myasthenia gravis. You can see it.
Who does?
The pastor of the Baptist Church is a mega church in Memphis. He has
He's got myasthenia gravis. Okay. So what's going to happen is you're going to get this autoimmune destruction of these receptors. That's going to look like facial paralysis. We don't get contraction. And so the patient is going to present with facial drooping and weakness in the face. You can actually do some tests that you'll learn later in the program to test for myasthenia gravis, because you can see maybe is this a stroke? Do they just have lots of Botox in their face, doesn't move? Who knows?
You said facial drooping and like
Muscle
Weakness,
contraction.
muscle weakness. So there's a couple of tests. There's one that we can do called the ice test. If you put some ice, this is not related to the class, but you can put ice on their face, you know, like rub it on their skin, and you're going to see if it makes it better or worse. Typically, that'll make it worse.
How would it be worse if we drink more or what?
Yeah,
Okay.
you'll learn about that later. You already did learn that now, but just so you guys know, there's a lot of cool things that we can do to test for that. Okay, so that is myasthenia gravis. Let's talk about botulism next. Botulism is going to be what you get from contaminated canned foods. That's the most common place in botulism. So, for example, if you were to like open a jar of spaghetti sauce or pasta sauce, normally when you open that can, you're going to get that pop, right? That's the seal letting you know that it's been broken. And now the top of it has that little bump that you can kind of push down. It's like, yeah. So the whole reason that exists is because Botox is caused by a bacteria that releases gas. So if you go to the store and you buy pasta sauce that has like a bulged lid, don't eat that one. It'll make you sick for sure.
you said it's botulism what?
Botulism. Botulism is the name of the condition.
Okay.
The toxin is called botulism toxin. Yeah, botulism toxin. It's released by a bacteria called Clostridium botulinum. You don't need to know that right now.
It's like microbiome.
I think that was in our
Yes,
last
it was.
semester.
You guys had micro last quarter?
Yeah.
We have it this quarter too.
Yeah, we have proper 1 and 2.
Did you get them from the get-go?
Yeah.
Oh,
Right out the gate.
okay.
How they have it set up is like we're all in lecture-based class right now.
Yeah.
And then apparently we're supposed to be getting into our labs like next quarter and fourth quarter.
Yeah.
Oh, that's way cool. That's way better. You might not think it, that's way better.
Okay.
We didn't get micro until like third quarter.
That's
Yeah,
crazy.
yeah. So no, that's way. How many micro classes do you have?
I think it's
Just
Two,
But
just
there's
2 right now.
bacteriology, parasitology or something.
I think there's a couple more.
We used to have 3 classes. It was parasitology, bacteriology, and then immunology.
Yeah, all
Yeah,
of
they
these
were-
are like
Who teaches those now?
little.
Okay, so it's not better. Well, okay, so we have botulinum toxin. So the way botulinum toxin will work is it is going to inhibit the release of these synaptic vesicles. So what's going to happen is it's going to basically erase step 3.
So it erase it, or it doesn't allow
It doesn't allow acetylcholine to be released. And so a lot of times if you eat, let's say, bad pasta sauce, you get infected with this botulinum, Clostridium botulinum bacteria, they start releasing the toxin and it starts slowly keeping your muscles from contracting. This can be a problem systemically because obviously we have to breathe. So if somebody gets botulism uncontrolled, their diaphragm will be paralyzed in a relaxed state, right? It'll just stay relaxed, it doesn't move, it can't contract. Nothing ever happens with it. And then you breathe. You die because you can't breathe. So that's concerning. Now, scientists, maybe 40 years ago, were like, this is a really cool idea. What if we capitalize on botulism toxin? And we stick it in a little syringe and we start injecting it into people's faces. Then their muscles can't contract. So that's why we get something called Botox. We just inject small amounts of botulism toxin into the facial muscles to keep them in a relaxed state, keep them from contracting.
Would that give you immunity against developing botulism?
No, just unfortunately not.
So in a sense, it just paralyzes the muscle.
Exactly. So it keeps them relaxed or paralyzed in a relaxed state. Compared to something like tetany or tetanus, which will paralyze the muscle, but in a contracted state.
And relax.
So tetanus will do the opposite. In tetanus, or lockjaw, what it used to be called, we release the acetylcholine, but we never break it down.
Gotcha.
Ideally, what we do is we send acetylcholine, it stimulates the muscle, and then we say, okay, that's enough. And then we break it down. We chop up all this acetylcholine, and then we recycle it so that the muscle will contract, and then it relaxes, and it's all good. But if we don't have acetylcholine esterase, the enzyme that breaks down acetylcholine, the muscle will contract and stay contracted. And so the person will also die because they can't breathe, but instead it will all be contracted versus in botulism they can't breathe, and it's because it's paralyzed in a relaxed state.
So it's tetanus one of our diseases? Or
That's one of the diseases. They used to teach it in the neurophys course. I'll teach it to you guys just so you have it. But yeah, so we've got myasthenia gravis, autoimmune destruction of the cholinergic receptors on the muscle side. We have botulism toxin, which inhibits the release of a neurotransmitter, acetylcholine, so the muscle stays relaxed. We have What was the other one? Tetanus.
Eaton.
Yeah,
Oh, we don't talk about that.
Lambert-Eaton. Yeah, that's one that I haven't really learned a whole lot, so I actually kind of just looked it up because they did not talk about that previously.
So what is I know it's like you get it from like rust or like whatever, like,
Use stuff on
function.
something.
Yeah, like what is it actually caused by? Like,
Tetanus?
Yeah, you said
Also
it's
by
the destruction of the.
yeah, it's also caused by a bacteria.
Okay.
I thought it would be bacteria.
Yeah.
That makes sense.
And so, like when people say like, "Oh, you stepped on a rusty nail," it's not necessarily the rust of the nail that's going to give you tetanus. It's that rusty nails tend to be in dirty, damp, swampy environments, right? You're going to be stepping on it in like the dirt.
Acetylcholine for calcium contraction.
Yeah, because you don't have calcium, you don't get acetylcholine release.
Okay.
Because calcium is what triggers the release of the neurotransmitter. So those are going to be your 4 neuromuscular junction disorders that we're going to talk about right now.
It seems like there's so much material on this exam, the midterm. Are we gonna, it looks like we're gonna have a prof session right before it then.
Yeah, there'll be one more next week. Because then next week we'll go over the autonomic nervous system. We'll go through it like
Really fast.
really well, and then we'll go over the different types of muscle fibers. We have like cardiac muscle, smooth muscle, and then some of the endocrine things related to that. Cool. I think that's probably where we'll call it good for the day. What questions do you guys have?